This recorded session is dedicated to our second module for Fundamentals of Zoology lecture. We'll talk about the cell and its biology, its biochemistry, and we'll give emphasis on subcellular structures, on organelles that make up or that are found within cells. Before we begin our discussion on the different classes and phylum of animals, okay, before we talk about their anatomy and physiology, it is important to ground ourselves on the basic unit of living systems, which is the cell. So, we begin by reminding ourselves of the basic tenets of the cell theory. Okay, uh, in our during our on-site discussion last Saturday, we talked about living systems and the characteristics, or the living organisms and the characteristics that they possess. And as we have established, okay, as of now, we only consider cell-based entities or systems to be living organisms, and those that are not made up of cells are considered as non-living organisms, or I mean non-living things. Okay. Now, a number of uh, there are a number of assumptions or tenets under the cell theory, but we'll just mention the most important ones. No? Number one, all living organisms, they may be composed of one or more cells. So there are living organisms that are unicellular, like bacteria. There are living organisms that are multicellular, like animals. The second important tenet of the cell theory is the cell is considered as the most basic unit of life. So that's already established, as I have shared with you last time. So all living organisms are only considered living if they're made up of cells, or if their most basic unit is the cell. And lastly, in relation to the biogenesis theory, or the biogenesis theory of life, all cells arise only from those that are already pre-existing. So has something to do with cell division, reproduction, and development. No? Now, so those are three important things that you have to remember when whenever you reminisce or look back at what the cell theory is all about. No? Now, there are two basic cell types for all living organisms. We have prokaryotic and eukaryotic. No? So prokaryotic is a cell type possessed or inherent in unicellular organisms like bacteria. Prokaryotic because or the term prokaryotic means before nucleus, pro, and then karyon means nucleus. If you look at the subcellular anatomy of a bacterial cell, you'll appreciate that it lacks nucleus. No? So it doesn't really have an identifiable nuclear region. Unlike eukaryotic cells that you can see here, which possesses a true nuclear region, a true nucleus. No? Animals, for the purpose of our course, belong or possess are essentially eukaryotic organisms because the cells that they possess in their body are possessing nuclei. Okay. Now, let's take a look at the generalized structure of an animal cell. Microscopically, if you look at an animal cell under the microscope, there are three um, visual parts, no? Very visual parts. Number one is the cell membrane. Okay, so let me highlight it. Okay, so the cell membrane, which is the outermost covering or the outermost structure of all cells. Okay, it separates the internal structures from the ambient, from the environment. And then there is the nucleus, of course, for eukaryotic organisms. The nucleus possesses or it encapsulates the genetic material of animal cells. And then there is the cytoplasm. Okay, so the cytoplasm, which is like an ocean of material, fluid like material surrounding all the internal subcellular structures, all the organelles, including the nucleus. Okay, so the cytoplasm, also called as the cytosol, or the possesses intracellular fluid, where the organelles are embedded in a way they're like floating in a sea of intracellular material. So three very visual structures in animal cells. So please remember these three. Okay. Then we have, let's take a look at the internal environment of animal cells. So we have here the nucleus, okay, possessing the genetic material. Within the nucleus can be subdivided into structures such as its own membrane, the nuclear membrane, okay, the nucleoplasm, which is similar to the cytoplasm but is located or localized inside the nucleus, and the nucleolus, okay, which is in most animal cells that are stained and if stained and to be observed under the microscope, you can appreciate the nucleolus as a very brightly staining structure. No? Now, in addition to or together with the cytoplasm, okay, the nucleus comprises, or together with the cytoplasm, these two structures, when they're put together, when you uh, consider them in a particular context, we call that the protoplasm of eukaryotic cells. Now, more on the structures that are very visual. Let's take a look at the cell membrane. So the cell membrane you can see here is a covering. So visually, it's the covering of all cells. For our case here, a eukaryotic cell with a flagellum, you can see the cell membrane. Also goes by the name plasma membrane. Okay. Let's take a look at the structure of the plasma membrane or the cell membrane. Okay. Um, Electron microscopy has allowed us to further study the very microscopic or submicroscopic cell membrane, and we can appreciate that it's actually a double layer, a double membrane. So it's composed of two layers that are sandwiched together. So this one and this one. They are they form they form a bilipid layer where different types of macromolecules are also embedded or are also in a way floating, okay, such as proteins that could be integral within the membrane, meaning these proteins traverse both sides of the cell membrane, the exterior and the interior. Also, some lipids like cholesterol molecules are embedded within the cell membrane. Carbohydrates, in conjunction or in combination with proteins, such as also called glycoproteins, can be found embedded in the cell membrane. Uh, what else? In relation or in association with the cell membrane, no, in the internal environment, in the cytoplasm, you can appreciate the presence of filamentous structures called cytoskeletal fibers that serve as the skeleton, no, of the cell. It's not a literal skeleton per se, but the cytoskeletal fibers or the cytoskeleton gives structure. No, it provides structure to the eukaryote or to a cell, so such that it doesn't really assume a two-dimensional state, rather a three-dimensional. It is assuming a three-dimensional state. So the cell essentially has a volume because of the shape maintained by the cytoskeletal fibers or the cytoskeleton. Okay. Also, before I forget, there are several theories in the past when we know less about the biochemistry, the biology of the cell membrane. Okay, there are several theories that relate to its character or to its nature. And the theory that is found to be the most plausible one is the fluid mosaic model, where you you treat or we treat the cell membrane as a dynamic structure, meaning um, it is in constant flux. So what do I mean with that? One structure, or let's say this integral protein here, okay, it can move from its original location. Let's say this is its original location, location A. Okay, after some time, it might move to another location, like location B. So fluid, meaning the components are moving. They're always in a constant state of flux. Okay. Then membrane components. Let's take a look at each of them. So the cell membrane is primarily made up of lipids. Okay, and these lipids are of different types: phospholipids, glycolipids, and cholesterol. No, so phospholipids are the major components. As you can as let's go back to the previous slide. Yeah. Okay, so each layer is composed mostly of phospholipids, right? So the gray structures, the gray colored small circles or spheres with two tails are phospholipids. Then we have glycolipids, which are lipids um, that are in combination with carbohydrates. And we have cholesterol, which is a pure lipid, important for the fluidity of the cell membrane. Then we have proteins, so membrane proteins, which can be integral or peripheral proteins. So integral proteins I mentioned a while ago are proteins that traverse both sides of the cell membrane or both environments, the extracellular and the intracellular, while peripheral, on the other hand, peripheral proteins are only present on one side, either the outer side, outermost, or the innermost side. Okay. So here are illustrations here are illustrations of phospholipid molecules. So you can see here how its conformation, okay, with tail of hydrocarbon tail of hydrocarbons, which are by nature non-polar. They repel water, okay, while the head, mostly in association with the phosphate group, okay, allows for the characteristic the characteristic um, by nature of phospholipid molecules. By nature meaning the head portion does not repel water. You know, that's capable of interacting with water, while the tail portion, the tails are water hating or water fearing, while the head is water loving or are capable of interacting with water. So one layer of phospholipids, okay, comprise one side that faces the ambient, and another layer of phospholipids comprise the other side that faces the internal environment of the cell. Okay, so yeah, I mentioned this already. So the heads are polar by nature, and the tails are non-polar. Now you might ask, if the heads of the phospholipid molecules are actually, or if the tails are non-polar, how is it that water can traverse the membrane, especially that water is a major constituent of cells. Water is a major constituent of living organisms. No? So later on, we'll discuss more about it. But essentially, there are several mechanisms that facilitate the transfer or the transport of water molecules from the internal environment to the ambient of the cell and vice versa. Okay. Now, let's take a look at the other components, the proteins, integral proteins, and peripheral proteins. Integral proteins are of different types. So there is a channel protein that allows the transfer of molecules from one side to the other. These channel proteins are as I meant as integral proteins. They traverse both sides of the cell, or both environments, and these channel proteins, uh, in terms of function, they allow specific molecules. So there are channel proteins for specific substances, specific molecules. Okay. Then we have carrier proteins. Okay. Carrier proteins, on the other hand, or I mean, similar with our channel proteins, also allow transport of molecules from one side of the cell, from one environment, or from the external to the internal, or vice versa. But mostly we associate carrier proteins to specific solutes, and these carrier proteins, for them to be activated, for them to carry out the transfer, there should be some conformational change in the molecule. Like it has a conformation that it receives the solute from one side, and later on will transfer it to the other side, and vice versa. So unlike channel proteins, carrier proteins must undergo conformational changes to facilitate molecule transfer. Okay. Then we have cell recognition proteins. These are integral proteins with identity markers, okay, with complex molecules that are attached to them, usually serving as receptors for other organisms, other unicellular organisms, or receptors for promoter substances that, once recognized by the cell to be present in the environment, will make the cell activated to do something. Like for the case of um, gene regulation. So these identity markers play important roles. Then we have receptor proteins. So these are also integral proteins with um, a section of them that allows the recognition of an external stimulus, or an external molecule. No? Somehow, you can say that receptor proteins and cell recognition proteins, they are both involved in in physiological processes that allow the cell to entertain signals from the environment. Okay. Then there are enzymatic proteins that can break down substances. these enzymatic or catalytic proteins can carry out substrate breakdown to facilitate a cascade or a signal, or to propagate a signal from the previous step or from its previous state. No? Okay. Then we have the cytoskeleton. So the cytoskeletal fibers, as I've shared a while ago, they are akin to the skeleton of the animal body, but they're not really like mineralized. No, they are proteins that allow for the cell to assume a particular shape, a shape that is usually or a shape that is dynamic, meaning it's not a static shape all the time. Okay, so these cytoskeletal fibers may attach to the cell membrane, may attach to molecules that are also embedded within the cytosol. Okay. Now, the cell membrane, more about it, there are some modifications that are present in different types of cells. Like for the case of cells that are found in the intestinal lining, there is microvilli, which are finger-like or hair-like structures on the apical portion of the cell, allowing for an increase in the surface area of that cell so that it can absorb material from the ambient. Then, for the case of prokaryotic cells, okay, a cell membrane modification is the glycocalyx. Okay, so this material or this covering, it's a gel-like material that is coating the outside of the outermost portion of the bacterial cell. It's compact and called a capsule in most cases. Uh, if the, if 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 it is more solid, we call it a capsule. But if it's not that solid, then we call it a slime layer. So the glycocalyx allows for an extra protection for prokaryotic cells, no, for bacterial cells. And for plants, a modification of the cell membrane is the cell wall, okay, which is made up of cellulose. Okay, cellulose is the primary polymeric carbohydrate that serves as the outermost structure of plant cells. So the green colored structure here is mostly the plant cell wall, allowing for the plant cell to maintain turgidity in most cases, or almost always the cell wall of plants allows for turgidity to be kept or to be maintained in the cell. Okay. Now we talk about the organelles for eukaryotic cells. Let's discuss the organelles, the subcellular structures that are found in eukaryotic cells. So let's first look at the nucleus. So the nucleus, within the nuclear compartment can be divided into different visual structures. So we have the nuclear envelope, nuclear membrane, which similar with the plasma membrane or the cell membrane, it's also a double lipid layer. No, the nuclear envelope is not a it's not a continuous structure in the sense that within the entire membrane, there are openings that allow for the exchange of material. So these openings called nuclear pores. Okay. Then we have the nucleolus, which is the primary concentration of protected material within the nuclear region. Then there are chromatin fibers that are embedded or that can be found within the nucleoplasm. Okay, so more about the nucleus, more about the nuclear compartment. So here you can see an electron microscopy micrograph of the nucleus. So the entire circle here or sphere is a nucleus, and you can see perforations within it. So those are the nuclear pores. These nuclear pores are actually protein channels that provide or allow for specific opening, or for that create these openings within the nuclear membrane. And as I mentioned, the nuclear envelope is phospholipid bilayer as well. Okay. Now, the genetic material is housed in the nuclear compartment, the DNA and the RNA. And DNA in all animals, or in all within cells, is actually highly compact. So, of course, it's a very long structure, it's a very long molecule, so it needs to be highly compact. So, it undergoes several layers or several stages of several stages of packaging, such that uh, it becomes a chromosome. And this chromosome contains millions of DNA bases. Now, aside from the genetic material, we can also appreciate in relation to, the genetic material being protected in the nuclear membrane, or I mean in the nuclear compartment. So we also have ribosomes. So ribosomes are structures that allow for the translation of proteins, allow for the interpretation of uh, um, the genetic code in order to produce or synthesize proteins. Okay. We'll not discuss more about, or we're not gonna dive deeper about protein translation, because that's not part of our module anymore. But just to introduce to you, there are subcellular structures called ribosomes that facilitate the interpretation of the genetic code for protein synthesis. Okay. Then we'll look into the endomembrane system. Okay, I think this is the slide. Okay, so I will pause here.